Thanks, Joe. So hi, everybody. Welcome to, I think we're at the fourth, might be the fifth in the seminar series. And really excited about today. We have two speakers today. First off, apologies for all the technical glitches. Maybe if we could move to the next slide. I have reviewed this in previous presentations, but just high level, we're really looking to get all the people together, as well as interested scientists, to really start to talk about effector function and the different things associated with effector function, whether you're interested in vaccines or monoclonal antibodies, or just in general how the cell's immune system interact. It's been a really enlightening seminar series so far. And so this has been put together on behalf of the Active Trace group. I co-chair that committee with Matt Hall, but we've brought together a number of people, if we go to the next slide, Courtney, to really look at two aspects of effector function. One is, and that's the focus of the group that's put these seminars together, is really translation of preclinical effector function to clinical COVID. The other group has really been looking at how to standardize effector function assays and how we then use that to inform how we move forward, whether that's on the vaccine front or monoclonal antibodies or any other therapy that might engage this aspect of the immune system. And so you can see the participants and facilitators here, really rock stars within the effector function world. And I've been learning a lot from each and every one of them. Unfortunately, Galit, who was supposed to be here to do the interactions, unfortunately had to send her regrets last minute. But I'm sure that she'll be listening to the recording as we move forward. And I think Trey put a link to the recordings in the chat. So maybe I could get started. Oh yes, the next seminar will be on February 1st from 2 to 3:30 p.m. Eastern time, where Galit Alter will be talking about polyclonal swarms and system serology, and Gester will be talking about Fc receptors and IgG allotype polymorphisms. It should also be a really interesting seminar series. But today we have Margie and Adam with us. And just to introduce Margie, I am going to introduce Adam. I'm going to do you guys at the same time, and then we can transition between the two. I think we could probably stop between, and I can't remember how much time we had allotted per seminar, but hopefully we'll have time, lots of time for questions. But Margie, amazing powerhouse in this field. She's an associate professor of engineering at the Thayer School of Engineering at Dartmouth College. Received her Ph.D. in molecular engineering from MIT and did her postdoc at Reagan. She's authored over 150 publications. I looked that up, Margie. And understanding the development of immunotherapeutics and vaccines, especially in the complex areas of HIV and now COVID. And Margie's going to look at antiviral functions of human antibodies. And if I could introduce Adam. So Adam is a professor of biochemistry, if you go to the next slide, Courtney, at South Dakota State, and the director of the NIH, I'm assuming I say COBRA?
Yep, COBRA.
Yeah, Bio Center Insights into Inflammation, which is a huge center looking into all different aspects of immunology. PhD in biophysics in FRET microscopy, super cool, at the University of Michigan. Completed his postdoc in phlegm microscopy and FRET algorithms at the University of Michigan, and did a senior postdoc in 4D FRET microscopy at Pasteur. And his lab's main research areas are really the growth, function, and differentiation of macrophages, and the development of new methods for imaging biochemical events within living cells. And so today, Adam's going to be talking about FcγR receptor signaling mechanisms in phagocytosis and antibody-mediated viral clearance by macrophages. So welcome to you both. Super interested in what you're going to be presenting today. Really looking forward to the discussions. Maybe I'll turn it over to you, Margie.
Thank you so much, Lisa and Joe. So let me get my screen shared. And I was given two titles. So I think that means I get twice as long as Adam. So I'll talk about antiviral functions of human antibodies with a focus on in vivo activities and highlighting some work from animal models that are currently used to explore the relevance and importance of these mechanisms of action. And I just wanted to start by noting that, there's a lot of emphasis on neutralization and evasion of neutralizing antibody responses in the context of SARS-CoV-2 variants, but neutralization in and of itself is an in vitro kind of artifact. That's not the real way pathogens are cleared or effectively neutralized in vivo. That couples to the Fc-dependent activities of antibodies, which can elicit antiviral or antipathogen activities of a diverse range of innate immune effector cells. So monocytes, neutrophils, other phagocytes, natural killer cells, induction of the complement cascade. All of these different effectors and effector mechanisms can contribute to the disposition of antibody opsonized infected cells or free virions. And so to remind people about what this really, looks like, this is a video of THP1 cells. So this is a monocytic cell line, and they are gobbling up the green fluorescent beads, which are coated with antibody on their surface, and pretty much ignoring the red fluorescent beads, which are identical on their surfaces, except for lacking antibody. And so these are really powerful activities that can complement or extend beyond neutralizing or direct antipathogen effects mediated by the variable domains of antibodies. And we know from many, many elegant experiments, I picked one classic one from the setting of HIV, that these functions are important in vivo. So in this study, a broadly neutralizing HIV-specific antibody B12 was passively transferred to rhesus macaques who were subsequently challenged with SHIV. And wild type antibody was able to protect almost all the animals from infection, whereas the neutralizing antibody mutated to lack binding to FcγR receptors and complement failed to protect half of the animals. So even a potent and broad neutralizing antibody in vivo to protect the host benefits from its effector function. And that's just one example. There are many. I picked some other examples throughout this talk that I hoped other speakers in the series wouldn't be alluding to. So this has also been observed in influenza. In the context of smallpox, when you measure neutralization, you actually don't see much unless you're performing that assay in the presence of complement. And even antitoxin antibodies, which, if anything, you would expect to not benefit from effector functions, even those neutralizing antitoxin specific antibody responses benefit from in vivo from antibody effector functions. So these activities are also variable, right? So what I've shown here is an illustration of the same antibody that's been subclass switched from human IgG1 to 2 to 3 and 4, and aglycosylated in black or afucosylated in light blue. And what you can see here in this phagocytosis assay, so a phagocytic score is reported on the y-axis, that there's a really strong dependence on this activity that's associated with the particular subclass and the glycan composition of a conserved glycan motif that's incorporated on the antibody Fc. And I think many people like to gloss over this complexity, especially the complexity that's not encoded in DNA. It's not templated. But here are a few examples of the impact of that little red triangle. So fucose, illustrated on the antibody glycan structure at the top left. And you can see that the presence or absence of that fucose has been reported to impact things as diverse as antibody-mediated organ transplant rejection, responses to and severity of COVID-19 illness. And my favorite example, because I've had enough little people in my life to raise an anti-red blood cell antibody response myself, presence or absence of that fucose can influence the severity of anti-allotype, anti-fetal antibody responses. And the last point I wanted to make about these functions is that they're tunable. And so the example I'm highlighting here comes from a pair of HIV vaccine regimens and trials. So RV144, or the Thai trial shown in red, you can see the number of subjects that mounted an IgG1, 2, 3, and 4 response illustrated in the bar graph, in comparison to the VAX-003 vaccine trial in blue. And what you'll notice right away is that VAX-003, which used the same protein immunogen as was used in the boosts in RV144, led to a dramatically different profile in the subclasses of antibodies that were induced by the regimen. So this is some evidence that there is an ability to control the types and the characteristics of the effector function responses elicited by immune exposures. But it's a really complicated landscape. So there are many activities and properties that will influence the in vivo efficacy of a polyclonal antibody response or the efficacy of monoclonal antibody therapies or cocktails. And so we pay a lot of attention to those that are Fab dependent and relate to antigen recognition and neutralization. But there's a whole host of downstream parameters, and biology is pretty efficient. So if each of these attributes were not important, and each of these effectors didn't contribute to robust survival of the host, we wouldn't have them, right? And so I think it's important to think about profiling and understanding the spectrum of responses, because they really are important in vivo. And we don't have to look very far, right? So here's a SARS-CoV-2 specific example that I think really drives home the value of in vivo neutralization, or in the collection of activities neutralizing and effector functions that antibodies elicit. So this was a set of experiments conducted by Kevin Saunders' group at Duke, where they isolated antibodies that enhanced infectivity in vitro assays. And they did that in either FcγR receptor dependent or independent ways. And they tested them for their effects in vivo. And what they observed was that in vitro, anti-neutralizing antibodies, so infectivity enhancing antibodies, were protective, right? So they provided complete or partial protection of the host, despite the fact that they not only had no neutralizing activity in vitro, but they actually exhibited an enhancement of infectivity in vitro assays. So I think if I were to have a thesis statement for the rest of my talk, it's this. So if you want to get breadth, you have to have breadth. And so that's a little silly. So what do I mean by that? I think we need to have antibodies that exhibit breadth and we have a spectrum of them present at different sites with different characteristics that will then exhibit a breadth of different functions. Having a response with a breadth of different functional attributes will help combat a breadth of different pathogens, including variants, and will also help make responses robust across person-to-person variability. Right? So I think if our goal is to have protective breadth against the face of a diversifying pathogen like SARS-CoV-2, we really need functional breadth, both in antigen and pathogen recognition, as well as in antiviral functions. And so I'm going to move to an example that I anticipated fewer people participating in this audience might be familiar with, which is neonatal herpes simplex virus infection. And so HSV is not a particularly severe disease infection for adults with normal immune systems, but it can be extremely consequential in early life when there are peripartum infections. But antibodies are tremendously protective. So the highest risk of transmission from in neonates is when mothers are infected, but not for long enough to become seropositive and to transfer their antibodies to the fetus. So those are the babies that upon birth are at the greatest risk of infection and the greatest risk of really serious and severe disease that even in the context of acyclovir antiviral therapies leaves many babies with significant lifelong morbidity. But then epidemiological evidence, the fact that seropositive women rarely, their babies rarely are affected, indicates how protective antibodies can be. And so in collaboration with the Lieb lab here at Dartmouth, we started to investigate the ability of monoclonal antibodies that target the viral entry mediator, glycoprotein D, and their ability to protect neonates in a mouse model from HSV infection. And I'll be talking about 3 different antibodies, CH42, which is a non-neutralizing antibody that recognizes the herpes virus entry mediator region shown in yellow, E317 or UB621 shown in blue, which recognizes the footprint that GD uses to interact with nectin, another cellular receptor binding receptor. And as well as HSV8. And so if you passively transfer any of these antibodies into a neonatal mouse, whether through their mom or directly administered to the pups will be resistant to succumbing to HSV infection. Right. So whereas pups treated with an IgG control, an isotype control, all succumb, there's up to complete survival of animals that are treated with any of the HSV specific monoclonal antibodies. And for the same dose of antibody, protection varied with respect to neutralization activity. So the most potently neutralizing antibody protected better than the antibody that didn't exhibit any in vitro neutralization activity at this dose. But if you knock out the ability of that non-neutralizing CH42 antibody shown in red to interact with Fc receptors or complement, and that's shown in the dotted line, this NA mutant, so it's a point mutation that eliminates the interaction between the monoclonal and these effector molecules. Now there's no benefit provided. Even if you pre-complex the virus with antibody in solution and challenge with that mixture. Similarly, if you repeat these experiments in mice that lack FcγR receptor expression, even the most protective neutralizing antibody is severely compromised in terms of its ability to protect against HSV mortality in this model. So you see the HSV8, which exhibited very complete protection in a wild type mouse, is protecting less than half of the animals when they lack FcγR receptor expression. So even the best antibodies lose almost all their in vivo efficacy in a host where effector functions are compromised. Waiting for my next slide. Right. This is also a dose-dependent effect. So here with a different, the same HSV-8 antibody, but a LALA-PG mutant of that antibody. So its Fc domain has been mutated to knock out both FcγR receptor and complement binding. With a low dose of antibody, there's really no protection, no survival advantage afforded by a potently neutralizing HSV-specific antibody. And if we swap strains, so all the prior data was collected with HSV-1 challenge, and if we substitute with an HSV-2 challenge, we really see that even at a high dose of antibody, without effector functions, a potently neutralizing antibody provides no benefit. Right? So in vivo activities can be quite distinct and broader than in vitro activities. And so from this HSV example, a breadth of functions certainly contribute to protection in this model. So both neutralization and effector functions, FcγR receptor dependent and complement dependent functions appear to contribute, and they help to provide broader protection in terms of the antibody doses that will protect the animals, the routes of challenge, and the routes of antibody expression or exposure, as well as varying breadth of protection across viral variants, so between HSV-1 and HSV-2. And this protection is against, I showed survival data, but it's against viral replication and neurological morbidities in adulthood as well. So what about SARS-CoV-2? So I wanted to give two examples, again, trying to pick things that other speakers hadn't already talked about, or I didn't anticipate future speakers in the series would mention. And I'll tell a short story about a MAB-like molecule that was a collaboration with Marzena Pazgir and a few other groups. And then some polyclonal responses in vaccinated and infected individuals. And so thinking through how to achieve functional breadth in the context of monoclonal and polyclonal responses, the way that Marzena approached this was to use an ACE2 Fc fusion and to engineer both the variable domain of this molecule, so the ACE2 receptor fragment, as well as the Fc domain. And so she's sequence modified, as well as had different lengths of the ACE2 extracellular domain, and introduced knockout and FcγR receptor binding enhancing mutations in the constant domain of this molecule. And I'll show just results from just one of the in vivo experiments reported in this study, which was a prophylactic experiment. So the ACE2 Fc fusion proteins were administered prior to challenge, and then viral replication was monitored using bioluminescent imaging. And what you can see here is that the wild type ACE2 Fc domain, M27, animals treated with that molecule showed greater viral replication than did those that were treated with the ghastly Fc receptor binding enhanced variant of this fusion protein. And that's the M81 construct shown at the right. And so in collection with other in vivo data and in vitro data in that paper, I can summarize it briefly just to say that both Fc and FV engineering really combined. They can reinforce each other to combine and enhance and make additive contributions to improving the breadth and potency of protection. And in this study, that's shown for death, for weight loss, for viral replication. It's shown across at least one viral variant and in therapeutic and prophylactic treatment settings. And so these two approaches of making the molecule be as potent as possible at both of its functional domains seems to have additive effect. And so if we think about how we might generalize some of these approaches, there's a wealth of antibody engineering strategies that can be used for this. So engineering antibody recognition for breadth and high affinity, and of course, neutralization, selection of antibody combinations or cocktails that might enhance multiple mechanisms of action, so neutralization or effector function, as well as exhibit complementary recognition patterns across viral sequence variants. And then lastly, the Fc domain can be modified by selecting the isotype, the subclass, the hinge composition, which impacts phagocytic activity, as well as the glycan incorporated on that conserved motif. Or as the example used, Marzena used primary amino acid sequence engineering to non-native engineered Fcs that have enhanced effector functions. So how about if we switch gears and move into the setting of polyclonal antibody responses? And I think we're all well acquainted with the difference between binding responses, which have tended to be better conserved across variants, than neutralization activity, which has been severely compromised in terms of its ability to port from wild type early Wuhan strains, for example, to Omicron. And so here's data for the particular cohort I'll be walking through today that shows for both vaccinated subjects and convalescent subjects, while there's good Wuhan neutralizing activity, there were almost no subjects that showed neutralization activity against Omicron. So neutralization is easily compromised. And what's shown at the right, though, are binding antibodies. And so these are at whole spike extracellular domain. In the top row, IgMs, in the middle, IgAs, and in the bottom, IgG, and these are serum antibodies. The vaccinated subjects shown in green, convalescent subjects in blue, and naive subjects illustrated in black across a panel of spike variants. And what you can see is that the binding antibody responses are conserved. It's not as though we see IgGs and A's and M's binding Wuhan and there's no cross-reactivity with Omicron. And if we summarize that binding data across the panel of variants, we can come up with breadth potency curves and breadth potency scores. And you can see that they differ by isotype depending on how the individual was exposed, whether that exposure to spike was through vaccination or natural infection. But there's definitely a breadth of antibodies that recognize viral glycoprotein. What does that mean for functions other than neutralization? So what's shown here is phagocytic responses, so ADCP in the top row, ADCC in the center row, using a measure of FcγR3a signaling as a readout, and complement deposition, ADCD in the bottom row of each graph, across spike variants on the left and receptor binding domain, RBD sequence variants on the right. And what you can see is that while there are differences in the functional potency of responses in vaccinated and convalescent subjects, particularly in the vaccinated subjects, functions are not completely lost as the virus diversifies. And so phagocytic activity, ADCC, and complement deposition are all reasonably well maintained even against the Omicron variant shown in orange at the far right of each panel. So functional breadth in terms of effector functions appears to be much greater than neutralization breadth. And if we summarize that data into similar breadth scores, as we did for the binding antibody data, it's certainly the case that greater breadth is observed for effector functions than neutralization for vaccinated subjects than infected subjects and against SARS-CoV-2 variants. also against emergent coronavirus strains, so SARS-CoV-1 on the left, and a little bit of activity against MERS-CoV-1 on the right, but not against endemic strains, interestingly, in which case the convalescent subjects really have elevated functional activity. So we think that really has to do with the conformation of antigen. It could also depend on route of exposure, but I think the use of stabilized antigen in the vaccine has some real differences in the immunogenicity and the antibody responses that are induced by those two different exposures. So if I were to summarize that data and marry it with the rich in vivo data for MABs and some clinical data from convalescent plasma from Andres Finzi's group, looking at correlates of protection for convalescent plasma, it really appears that FC and FV breadth combine to contribute to the breadth and degree of protection for polyclonal antibody responses as well. And so how can we make polyclonal antibody responses better? I think that's an open question that we have some empirical insights into, right? So immunogen design, I mentioned those differences in the breadth profiles observed between vaccinated subjects and naturally infected subjects. So stabilizing the spike protein seems to potentially have had an effect on domain and epitope specificity that influences the breadth of effector functions. Vaccine design, so maybe it's the route of exposure and the immune compartments that are presented with antigen influence the isotype distribution. So we saw IgM and IgA responses being higher or comparable in natural infection and vaccination, whereas IgG responses were clearly elevated among vaccinees. Other experiments have elegantly shown antigen dose and persistence, so the timing longitudinally of antigen exposure influence response magnitudes and characteristics. And the number of boosters and the composition of the vaccine can certainly influence IgG subclass glycosylation. And I think we'll hear more about those concepts from later speakers. Similarly, the adjuvant or the natural adjuvanting capacity of the vaccine delivery or the pathogen itself will elicit different inflammatory signals that are also thought to be part of what plays a role in influencing or driving the antibody response magnitude and FC characteristics. But there's no magic recipe. So I think this is still an area of active investigation. You know, what can be done if we know the types of antibodies that are associated with the best protection? How do we induce the immune system to express a response with those precise characteristics? So in summary, effector functions often contribute in vivo. And the composition and the relative importance of different mechanisms of action probably differ depending on the context. So whether you're talking about prevention or therapy, the site of action or the site of exposure, whether it's mucosal or systemic, the virus being recognized. So an archival strain versus an emergent variant, as well as across the breadth of variable domains. And so open questions, you know, I talked about how much control we have as vaccine designers on influencing this, and I think the answer is less than we'd like, but there's evidence from comparative studies that suggest if we figure out the rules, we can leverage these aspects of biology. And I think, you know, an open question as to how to accelerate clinical translation of FC modifications that improve effector function is another area that I think maybe I've been surprised it's progressed. It hasn't progressed faster. And that's because, you know, if we look to HIV for an example, and I think this is the last slide, we can see many antibodies whose activity in both mouse models and non-human primates is improved by having effector functions. Those are all shown in green, and relatively few examples shown in red where knocking out effector function reduces in vivo activity. But despite the maturity of the Fc sequence engineering efforts that have targeted enhanced effector function, there's much more limited evidence as to their importance. Excellent evidence in mice. So here I've just given a couple of, again, examples limited to HIV, where for a diversity of antibody specificities and clones, you can introduce mutations and improve efficacy. But when this has been tested in non-human primates, it's definitely been less clear that these mutations will improve activity. Although I think the day is coming where we'll see more such reports. And promisingly, some of those variants are now being translated clinically. And so with that, I'd like to thank my collaborators and the graduate students who contributed to the data that I shared today.
Fantastic, Margie. That was a really awesome overview of effector function. I'm not seeing any questions coming through in the chat. I'm not sure if there are any maybe clarifying questions, and then maybe we could jump to Adam's presentation if there aren't any, and then sort of have a discussion at the end. Does that sound okay with you?
That sounds great.
All right. Doesn't look like there's any. You were so clear. I thought you were really clear as well. That was really amazing. So maybe we'll jump over to Adam. And he's going second because he's talking about macrophages, so he would have swallowed up all the time otherwise. Get it. So Adam, the floor is yours. Hopefully you can project your slides, and keep the questions coming in the chat if you have any, or you could direct them directly to me if you didn't want to put them in the general chat as well.
Okay, just to check, can you see my slides?
That's a beautiful and very scary picture. Thank you.
Excellent. Oops, let me back up one here. If I can, let me hide something here. Okay, super. Now I can see my slides, even better. Okay, well, first off, thank you very much for inviting me to give this talk. And I've got to say, following Margie, that's a tough act to follow. What I want to do today is talk about the role of the macrophage and Fc receptor function in the macrophage, and just to try to lay down a few ideas. Some of them might be right, some of them might be complete speculation, but at least to get folks thinking about how the macrophage may be functioning in viral diseases. What you're looking at here in MESH is a bone marrow derived macrophage from a mouse. And the magenta color is a B cell that is labeled with or decorated with anti-CD20 antibody shown in magenta, and you'll get to see a movie from this later. But this really is what our lab studies, is how do phagocytes recognize antibodies and then from that mount immune responses. Okay. So in the context, and what I'm going to try to do today is take sort of our knowledge about macrophage function and Fc receptor activity and try to bring it towards COVID. Much of what I'll be drawing heavily on other labs' publications, as well as some of our own ideas to try to paint a little bit of a picture about this. So in COVID and many other viral respiratory infections, alveolitis is one of the major pathological hallmarks. That is inflammation of the alveoli, the region in the lung where the gas exchange occurs. And this inflammation of these alveoli is characterized by activation of macrophages, recruitment of neutrophils, permeability of the vasculature near the alveolus, activation of platelets, and so on and so forth. That in the context of diseases such as COVID-19 and influenza can drive much of the pathology leading to severe disease. So as many of the speakers in this series have already talked about, antibodies can convey outstanding protection against lethal disease and respiratory infections. And in an ideal sense, antibodies can act as I've shown here, they can provide neutralization, that is, prevent the virus from infecting the target cell and basically leave the patient completely asymptomatic. I'll point out that in part of that is these opsonized virions have to go somewhere. And what I want to impress upon you today is they're often going to be cleared by phagocytes. And the macrophage is one of the main cell types that's going to mediate that clearance. Okay. In the absence of complete neutralization or partial neutralization, these viruses can infect certain cell types, and those cell types can then become sources of new virions, but also can be recognized by antibodies through displaying viral components on their surface or alterations in the cell as they undergo cell illness, if you will, during the viral infection. Antibodies can recognize those cells, those infected cells, and trigger all types of, or a number of types of responses, including activation of macrophages through antibody-dependent cellular phagocytosis, which we'll be talking about extensively today, complement deposition, and complement cytotoxicity, as well as natural killer cell function. Okay. And so what I want to do today is walk through particularly these ideas with regard to the role of the macrophage in these various processes. Now, one of the first things I want to impress upon you is macrophages are sort of the vacuum cleaners. We used to think of them as the janitorial cells of the body, but of course they have profound immune functions. But I want to remind everyone that these cells sit in effectively all of the tissues, and they contribute to clearance of debris as well as pathogens from that tissue. That function, however, can operate in sort of two modes. One, a non-specific mode, and I'm showing you a video here of macrophagocytosis. This is a macrophage that is, you can see these fluid phase vacuoles appearing. This is a macrophage that is gulping fluid through the process of macrophagocytosis and basically filtering its surrounding environment. Eventually, the material that's brought in into those macrophagosomes will be degraded by fusion with the lysosome. This process is running constitutively in many parts of the body. For example, this is a microscope image of the mouse ear skin following injection of a fluorescent dextran. And you can see these little stellite patches. These are the macrophages lining a blood vessel that have taken up that dextran through this process of macrophagocytosis. Additionally, though, receptor-mediated uptake, such as through FcγR receptors, can greatly facilitate uptake of particles, small particles, including viruses. And what I'm pointing to over here is an example in red of virion-like size particles. They're actually liposomes coated with an antibody, IgG2A, and very efficiently taken up into small endosomes. And I've also contrasted that against dextran, which is taken up by macrophagocytosis. Importantly, the dextran is at a very high concentration, as opposed to the uptake of these virions through an antibody-dependent, receptor-dependent uptake, which can be in which macrophages can capture even single virions very efficiently through this process. Okay. And there's 2 points I'm claiming here. Antibody-dependent viral uptake is mild and kind of a low or no inflammatory reaction, and I'll talk a little bit more about that now. So the macrophage can perceive antibody on targets through activating Fc receptors, which I've shown some here. I'm not going to get into too much of the specifics of these, other than there are very high affinity receptors for certain subclasses of IgG and varying affinities, I should say the affinities of these vary depending on the subclasses of IgG. All of them can signal through spleen kinase. So phosphorylation of these receptors activates that kinase, and it is recruited to these receptors. So the recruitment of Syk kinase to the Fc receptor is an indicator of its activity. There are also inhibitory receptors on the surface of the macrophage that can antagonize Syk's signaling, including the FcγR receptor 2b, which can recruit SHIP1, the lipid phosphatase, and SERP alpha, which can recognize CD47, basically an innate don't eat me self signal present on host cells. And that receptor can recruit the protein phosphatase SHIP1 to antagonize Syk kinase's function. Just as an example of the recruitment of Syk, this is now I'm going to show you a movie of a macrophage engaging a supported lipid bilayer displaying IgG, which is labeled with a fluorescent dye in magenta, and the recruitment of Syk. And so this is sort of like the macrophage trying to eat the glass coming at us view. And you can see the recruitment of Syk to these Fc receptors and the microclusters that form. Importantly, the clustering of the Fc receptors is central to its activation and downstream signaling. And I want to talk a little bit about how that clustering is important for viral infections. Okay, so we can think of different contexts for activation of these Fc receptors where even a single antibody decorating a virion could dock to an Fc receptor, like particularly the high affinity receptor, FcγR1, and that could mediate endocytosis of that virion and its clearance. I'm calling this passenger endocytosis because in this case, a single antibody is not sufficient, I'll show you some data in a second, to trigger activation of these Fc receptors. So this would be just riding in through a passive endocytic process, but it would aid in capturing the virion. We think that approximately 5 IgGs, so 5 Fc domains, clustering, I've got 3 receptors shown here, but I guess 5 receptors, would be sufficient to activate Syk kinase and trigger rapid endocytosis, as well as some additional signal transduction from endosomes. Okay, but we think of this as sort of a modest or a small signaling process. If we start to aggregate now IgG into larger patches and cluster more and more Fc receptors, so we'll call that polyvalent aggregation, so IgG is in excess of 20 or so. This will lead to prolonged signaling at the cell surface, as well as signaling from larger compartments, i.e. phagosomes. And this is where the cytokine response starts to become much larger. And this is also the basis for, if you're running IgG serum assays, activation assays for macrophages, usually why we use plate-bound IgG and then react macrophages onto those surfaces, because this is where you're going to have your maximum engagement of Fc receptors and signaling. Okay, so the point I'm trying to impress upon you here is that the number and configuration of IgG Fc receptor interactions shapes the response. And I'm just highlighting some work in collaboration with Momenta, which is now the company now with Johnson & Johnson, and their work on developing this molecule right here called CYF1, which is an Fc receptor inhibitor. And I want to use this work because it points out it highlights much of what I said regarding valency and activation of Fc receptors. So if we have, these are basically antibodies that lack the FV portion and are Fc only. So single Fc will dock to the surface of a THP1 cell. It will not recruit Syk kinase. Trimeric CYF1 or 3Y Fc will also dock to the surface of this is a macrophage-like cell, but it will not recruit Syk kinase. Pent-X or a 5 Fc engineered molecule will dock on the surface, rapidly recruit Syk, and quickly be packaged into endosomes. Okay, and so this is where that number of about 5 IgGs should be sufficient to drive FcγR activation and endocytosis. Whereas a polyvalent multimer, this will reside on the surface of the cell for considerably longer periods of time, and will also recruit Syk kinase. So we think of 5X as being sort of the threshold, 5 Fcs localized into a patch, to get activation of Fc receptors, larger multimers facilitating activation of increasing numbers of Fc receptors. And I think that data lines up pretty well with this paper here. I've got the citation over here, describing what happens in the context of opsonized SARS-CoV-2 particles. So in the case of viruses, very minimal cytokine activity when opsonized with serum or specific anti-spike antibodies. However, if the spike protein is presented on the surface of a cell, antibodies against that can give a very small cytokine response. So consistent with everything I was just saying, that ligation of a small number of Fc receptors will give a very small cytokine response, downstream signaling response. However, if used in conjunction with other agonists like poly-IC, a TLR agonist, that can greatly enhance the cytokine reaction that's stemming downstream from Fc receptor activation. And indeed, in this study, they actually used plate-bound anti-spike antibodies, or spike-on-plate anti-spike antibodies. And the only cytokine that was produced even in that context is a little bit of IL-8, so a neutrophil chemokine. The big nasties, IL-1 beta, IL-6, TNF-alpha, these required a co-stimulation with poly-IC. So I guess what I'm driving at here is, again, reminder that configuration of the FCs and their presentation will shape the strength of the signal. And then I'm also introducing the idea here that additional signals, immune activating signals, are required to have very strong cytokine responses. Okay, so I guess what I'm doing is I'm kind of grading the immune response in FcγR function through the lens of the macrophage. So again, ideal neutralization and simple clearance of the virion, that would be asymptomatic, no problem. Maybe next best is a scenario in which, and I'm going to try to make this case now, that the macrophage can engage with the infected cell and perhaps in conjunction with natural killer cell activity, lead to clearance of these infected cells. But this may require some reinforcement of a few additional effectors, such as additional monocytes or K cells. And that's in contrast to the Fc function, which is going to be more problematic when you have large quantities of infected cells, more than can be handled by this middle scenario. And that's where we're going to require T cell help to potentiate the macrophage activity, as well as additional some of the big nasty cytokines, which are going to drive heavy neutrophil recruitment and activation, as well as NK cell activity and cell lysis. Okay, and certainly complement deposition. Okay, so sorry, Margie, I do have one of the same papers you pointed towards. So we know that Fc receptors are very important and can provide protection for viral infections. So influenzas, going back to 2001, this beautiful experiment from Victor Huber, looking at lethal influenza challenge in mice, and comparing FcγR gamma positive and FcγR gamma negative mice. In mice, all of the activating signals signal through the activating chain, the gamma chain. And so if you're lacking the gamma chain, you have no activating FcγR function. And what they found in this work was that that protection in these H1N1 vaccinated mice required activity through the Fc receptor, and they concluded that phagocytosis makes a significant contribution to protection. Now, importantly, this isn't the same as having a perfect neutralization where you'd be asymptomatic. These mice do become ill over time. They lose body weight, but they recover, and they recover if they have FcγR function. Similarly, anti-stock antibodies, broad neutralizing antibodies, here shown in this experiment here, do not completely neutralize against infection, but require FcγR function, and particularly require in IgG2A, which in mice mediates, we think, is the most potent mediator of phagocytosis, as well as potential other Fc functions for protection here. Again, these mice become ill, but will recover. Okay. So, so if we think about the macrophage function during viral infection, I'm going to give you some examples of some of the things that I think are important here. And much of these ideas come from the therapeutic antibody field, in which we're generally trying to remove specific cells, cancer cells, from the body. So we know from that space that highly effective antibodies like anti-CD20 antibodies can drive the clearance of B cells, B cell lymphomas, aberrant B cells, similarly with T cells and other lymphocytes, and that this is a highly effective process, and it's immunologically quite calm. So it has minor extent of inflammation. So, for example, patients that are being treated for chronic lymphoblastic leukemia or other B lymphoma type disorders will receive infusions of these antibodies. It's generally an outpatient procedure. They may feel a little tired, but generally don't have too significant of a reaction. Similarly, in mice, we can repeatedly dose mice. This is actually data from Mark Craig and Stephen Beer's labs, showing repeated dosing of mice and depletion of B cells over time. Don't worry about the transgenic, but these mice are generally running around looking very normal, behaving very normal, and having modest inflammation. Now, what I don't want to say is that there's nothing going on here. There's actually, there's definitely a cellular response. So I'm going to, over here, we have an example of a mouse in which we're using a glass microneedle to deliver IgG-coated erythrocytes under the ear skin, and then imaging with a 2-photon microscope in collaboration with Dr. Darcy Fink here at SDSU. And what you can see is this is done in the MAC green mouse, so the macrophages are green. You can see red endosomal contents. Those are the erythrocytes, which have been already phagocytized and degraded. This is 6 hours post delivery. And the other thing that you can see in this movie is the recruitment of additional monocytes to the site. We have a blood vessel running through the middle here. So I guess the point being that there is some inflammation. You have some recruitment of monocytes, but if you look at the gross morphology of the dermis in these mice, they resolve this within 24 to 48 hours. So we need to use very careful fiducials to even find the site of the injection. So the inflammation here is very transient and very quickly resolving, despite all this activity that you can see with the recruitment of additional effectors. Okay. Now, if we think about this in the context of a respiratory disease, the situation is a little different than I showed in the dermis. And that is that the number of macrophages available to help clear the infection is far fewer. So this is some imaging data from the Cubes lab showing the distribution of alveolar macrophages, shown in green, migrating around the alveoli in a living mouse. And one of the key findings from this work was that there's less than one macrophage per alveoli. Less than one alveolar macrophage per alveoli. Now, there's also additional interstitial lung macrophages and neutrophils that are migrating through this space. But in terms of the cells that are going to interact with those type 2 pneumocytes that become infected during COVID, the patrolling of the alveolus is actually fewer than, you've got less than one cot per alveoli, right? So these macrophages have to migrate around. So I think one of the questions that this raises is, perhaps there aren't enough macrophages to be able to deal with viral clearance or clearance of infected cell, even in the presence of IgG. So they're going to have to recruit effectors similarly to what additional effectors, similarly to what I showed in that previous video. So they're going to have to bring in additional monocytes to replenish and maintain their function in that context. This also raises the question as to whether or not these alveolar macrophages could phagocytize a type 2 pneumocyote. Okay, and I'll point to a little data on that in a second. So now I'm going to switch over to talking about phagocytosis of cells, of host cells or infected cells. Okay, so everything I've told you about the Fc receptors so far applies here as well. In this case, however, the target cell presents would have the antigen, the antibody on it, which would trigger clustering of the Fc receptors, initially recruiting Lin kinase or other Sarc family member kinases, and then subsequently Syk kinase, which will amplify phosphorylation, drive multiple downstream signals that regulate actin polymerization and engulfment of that target or infected cell. Okay, so what does this look like? Now you get to see the cool movie. Here's this macrophage with this mesh in mesh, and what you're going to observe now is, in this case, a B lymphoma cell dropping into the field of view and engaging with that, being engaged by that macrophage. So that is the antibody in magenta clustering at the interface, and then large-scale actin rearrangements leading to the construction of the phagosome and the engulfment of that lymphoma cell. And I think the point here is that the clustering and the size of those patches, again, is very important for generating a sufficient signal for this macrophage to engulf and internalize that target cell. And then the second point is that for a free-floating or weakly attached lymphoma cell, we think of phagocytosis or ADCP as being a fairly efficient process for leading to the clearance of those cells. Now, what about cells that are adherent, like these pneumocytes, right? That are these epithelial cells that are in contact with their neighbors and in contact with extracellular matrix. I'm going to point to a very recent paper. It's currently in bioRxiv. I think it's about to come out from the Dennis Deischer lab, in which they've made, they're looking at a tumoroid model, and then also studying this tumoroid model in mice. So they're growing lymphoma cells in aggregate. And what this paper shows is that, and then using a therapeutic antibody to drive macrophage responses against this solid tumor. So these images here in magenta are macrophages, and in green are the melanoma cells. In the tumoroids, in the absence of antibody, the macrophages will simply interdigitate inside of the tumoroid, but in the presence of an anti-TRP1 antibody, that's a surface antigen on the B16 cells, and in combination with CD47 blockade, these macrophages will cooperatively maneuver around within the tumoroid, eventually engulfing and destroying these B16 cells. Now, importantly, unlike the movie I just showed you, where a single macrophage could take care of the do the job, here it's the thinking of the authors of this work believe that these macrophages actually have to work cooperatively. So I guess what I'm trying to do is reinforce this point that likely you're going to need to recruit additional effectors, additional monocytes differentiating into macrophages, and have some additional help to be able to deal with to be able to mediate the antibody-driven response against a more solid or anchored solid tumor, or in this case, a more anchored cell like the infected pneumocyte. So now I want to talk a little bit about what are things that can make ADCP better. And this is the main focus of my laboratory, and in collaboration with Mark Craig, who you'll hear from later in this seminar series. So when we think about what can potentiate Fc receptor function, I'm going to point to the interferons. There's a large swath of evidence now that interferons acting on macrophages can dramatically enhance their antibody-dependent functions. In particular, so both type 1 and type 2 interferons, with sort of maybe the classic example would be your Th1 response from a CD4 T cell producing interferon gamma, or potentially even NK cells producing interferon gamma, and type 1 interferons originating from the virally infected cells and in the immune environment. So we've been conducting sequencing experiments, looking at different macrophage activation states and identifying gene modules that are associated with potentiation of ADCP. And so as you can see, they're very large. This is the 2,000-foot view here of different claves of genes that are upregulated and downregulated in response to various stimuli here, and particularly interferon alpha and interferon gamma. Both are doing clearly different things to the macrophage. Okay. So if we look simply at Fc receptor expression, we can see that this is done from RNA-seq and from flow cytometry data, that the Fc receptor distribution here on the surface of mouse macrophages is heavily influenced by the interferon environment that these macrophages are experiencing. So M0 would be what we think of as our tissue or homeostatic macrophage, and type 1 interferon, or interferon alpha and interferon gamma driving significant upregulations, as indicated by the arrows. Most notably, interferon alpha and interferon gamma driving significant increases in expression of FcγR1, the high affinity Fc receptor, and FcγR4, also an intermediate affinity receptor. So one way that interferons make macrophages more potent eaters is by upregulating Fc receptors on their surface. Okay, we're also using CRISPR to try and identify functional correlates. So this is an example of a phagocytosis assay in our lab using erythrocytes coated with different types of IgG subclasses, and then macrophages bearing different FcγR knockouts. We're able to quantify and then rank their functions accordingly. And we can then apply that to address and test questions about how important these interferons are, or other activators such as LPS, in driving phagocytic responses. So what's being shown here on the y-axis is the number of red blood cell targets eaten per macrophage as a function of IgG concentration. As you can see, both LPS and type 1 interferon dramatically enhance phagocytosis over a M0 resting cell. However, if we knock out FcγR1, we can see that at this low density that FcγR1 expression is the primary driver of this response. And likewise, we can knock out the interferon alpha receptor and demonstrate that interferon alpha is specifically working through its cognate receptor. LPS is obviously working through a different receptor. Okay. We've broadened this approach, and we are using CRISPR whole genome screening now, in which I'll just briefly describe how this works, to identify different claves of machinery that can facilitate or inhibit macrophage Fc-dependent functions. So what we do in this case is we harvest bone marrow from Cas9 mice. We can transduce them with a lentivirus to deliver a guide RNA. That guide RNA can then target Cas9 to make a gene disruption such that each macrophage bears a different gene disruption. But the lentivirus integrates that guide RNA into the host cell genome. We can then challenge these targets with antibody-coated particles, sort them out using flow cytometry, given that the particles are fluorescent, and group them into high and low function groups. From that, we can then look at frequencies as to the number. We then do deep sequencing, to quantify the number of guide RNAs that are represented in each of those groups, high and low, as well as the pre-challenge and input fractions. And from that, we can then rank, capture the frequencies from them. So for example, this guide here, we have 4 guides here targeting this gene, WASF2. We can see that these cells are depleted in the high function group, enriched in the low function group, and neutral in pre and input. So we conclude that WASF2 is important for the phagocytic function. This other gene, MRC2, is flat across all categories. In terms of frequency, we can conclude that it's not important. From that, we can rank all genes in the genome, and then use that to infer their relative importance for a particular antibody-dependent phagocytic function. And we've been doing this across this is just a small snapshot of a handful of the immune modulatory conditions we've been looking at, but we can begin to define claves of genes that are supporting different attributes or aspects of these antibody-dependent functions. So one that came up, and I think it may be important in the context of these viral infections, but hasn't received a lot of attention, is the reactive oxygen species. So antibodies activating Fc receptors can drive the NADPH oxidase, which is this machine down here. We typically associate that with an antimicrobial response. However, it is also important for chemotaxis and recruitment of additional effectors, and can be a potent driver of inflammation in tissue. So our CRISPR screens, in the context of interferon gamma activated macrophages, identify all of the known components of the NADPH oxidase as a highly important aspect of downstream activation of Fc receptors. So I just want to highlight that concept, and that may be important in phagocytosis. So summing up what I've just told you, antibody-dependent cellular phagocytosis, antibody-dependent reactive oxygen enhancement, it can be driven by interferons, both type 1 interferons, which primarily drive expression of FcγR1, and interferon gamma, which can drive FcγR1 and FcγR4 expression, down modulate SIRP alpha expression, and increase activity of the NADPH oxidase. So anyway, we have primarily been using these screens in the context of trying to enhance antibody functions for more oncology-driven or cell-depleting lines of research, but I think they'd be very useful for also looking at infectious disease. And also we're trying to infer some of the ideas from infectious disease. Okay, I'm close to summing up here. The last thing I want to point to is another aspect of macrophage function in the context of respiratory diseases, and that's sort of the ugly. So if you have activation of complement and significant amounts of cell lysis, but insufficient numbers of phagocytes to remove that debris, you can have an accumulation of lytic debris from these cells. And that lytic debris can include all kinds of things like damage-associated molecular patterns, pathogen-associated molecular patterns, ATP, which can then activate potently stimulate macrophages into a very inflammatory phenotype, and potentially drive pyroptosis, which is a very specialized form of cell death, in which certain pores, the gasdermin D pore, can assemble on a macrophage and allow maturation and export of IL-1 beta, IL-18, COX-2, very potent inflammatory cytokines. So macrophage, so there's been a number of very interesting studies about the role of the macrophage in alveolitis and COVID-19. I just want to nod to these briefly. One, looking at human autopsy data, the other one in sequencing of cells from ball fluid, as well as a humanized mouse model in which the mouse, it basically has all of the myeloid cells are human. And these two studies agree that macrophages, particularly the alveolar macrophages, can become infected by the SARS-CoV-2 virus, and that infection leads to a very inflammatory function of these cells, recruiting massive amounts of additional immune effectors and driving the alveolitis in severe COVID. Now, I think one of the challenges here is determining whether, how do we really know that these macrophages are infected? They, as I've just described, they're very efficient at constantly eating other cells and eating debris in their environment. So one of the challenges is if you see viral material inside of the macrophage, is it because the macrophage was actually infected, or is it simply because it phagocytize that material. So that's, I think, one of the challenges facing our field right now is understanding exactly what we mean by macrophage infection versus phagocytic uptake of infected cells. This paper here from the Flavelle lab was particularly careful about this, looking at viral RNA, double-stranded RNA, which would indicate replication in these cells as well. So it looks like there's some potential for maybe a subset of these macrophages to become infected. Do antibodies help or inhibit this process? Again, we're back to that question of how neutralizing they are, or can they potentially convey some infection? But I'm going to kind of take the perspective of, I'm not really sure that matters for the disease pathology. Okay, in the sense that a subset of these macrophages may actually get infected, but again, as I pointed out, many additional immune effectors have to be recruited to support that phagocytic clearance. And those, I don't think there's any evidence in SARS-CoV-2 that those additional cells are also becoming infected. Okay, so I'm going to sum up here. What I hope I've done for you today is giving you a perspective through the macrophage lens of how antibody Fc function can contribute to viral clearance and prevention of illness in an ideal case, highly neutralizing antibodies. How it could potentially, so Fc-dependent functions can also lead to protection and may contribute to resolution from contributions by the macrophage, and as well as the more problematic case where partial macrophage functions may involve excessive inflammation and drive some of the pathologies associated with the disease. And with that, I'd like to thank my lab, people that have contributed to the work over here, as well as our collaborators at Southampton, and our funding sources from the National Institutes of Health and the National Science Foundation. With that, take any questions.
Thanks, Adam. Amazing microscopy throughout that presentation. That's pretty impressive. So I've got, oh, and there's some more questions coming through. I had one that came directly to me. I don't know, Margie, if you wanted to put your video back on. I guess the first question from Matthias in the chat, I'm not sure if you noticed it, I might not read through it, but talking about LLPG and some of the LLA mutations on some of the neutralizing antibodies, and how we sort of come to grips with the differing results from there, whether effector function is needed or not, and what are the factors associated with it.
Sure. Yeah. So, I mean, I think for non-human primate data, the tests are 5 to 1, right? So 5 different antibodies or bispecifics that have demonstrated contributions of effector function, and 1 PGT121 that does not. And that one super bothers me. It doesn't bother me that an antibody would exist that doesn't benefit from effector function. But what bothers me is that a different result was observed when that same antibody was tested in mice. And I would love to know why, if it's a model-dependent answer, if it's a challenge strain or route or condition associated answer. But I don't think that's clear yet. And I think that's an example of why, when I was sitting here trying to compile content for the talk, I really came up with breadth is important, right? So not all the antibodies are going to be the same. Even if you're really confident about how an antibody works in a given model, there's probably another variable where that balance will shift. And that variable might be totally out of your control, such as host genetics. And humans are pretty diverse. So I think, there's a lot of evidence that suggests we can't write off any of these activities if we want to have optimally protective vaccines and monoclonals.
And I guess the other wild card here is that for some of these, you're using NHPs, and the affinity of a human FC, if sometimes that wasn't used, but sometimes it was for an Fc gamma receptor in an NHP model might be very different from human, and of course from mouse. Thank you. This came through directly to me for a question for you, Adam. It was in reference to your story around the pentamers with the sick signaling you were showing, and wondering about whether or not, I guess in general, can there be too much signaling?
Yeah, so is there like a super agonist for FC receptors? That, I think, is a great question. So I think that's sort of our plate-based assay, right?
Mm-hmm.
So frustrated phagocytosis, laying antibody down on the plate, dropping macrophages onto that. They're unable to endocytose it. They're unable to phagocytose the plate. And so you have a constitutive signal there. But even in that context, so that example I showed in that from the paper on SARS-CoV-2, even in that context, though, generally you have to have some additional stimulus, that this other TLR agonist, additional interferons to get a very dramatic cytokine response out of a macrophage. So certainly, I think there's a large gradation of signals here, and I think, and also I should point out, I'm talking macrophage specific lens. This also is going to matter if we're talking about other cell types like neutrophils, right? Which can have a very violent reaction to extensive FC activation, such as netosis and pyroptotic death.
Yeah, I was struck. I hadn't actually ever heard the statistic of having less than one alveolar macrophage per alveoli before, but I guess it kind of makes sense. It's kind of interesting to know how that changes as you age, right?
Mm-hmm.
Or if it does. Another question from Peter for you, Adam, in terms of the, and he had mentioned similar to BCR microclustering for B cells, is it possible that FC receptor clustering on MACs or other EPCs could shuttle antigen to the endolysosome for the MHC processing?
So very, very likely. And I think much of what I said will have partial truths as well for dendritic cells in antigen presentation. There is some very interesting data in that paper from Dennis Deischer's group that I was referring to, where they're treating these melanoma tumors in mice with therapeutic antibodies, and they're looking at the vaccinal response in that context. So I would point you to that paper for some great insight into this. But absolutely, portions of these phagocytic targets are going to be presented on class 2, and certainly the immune environment surrounding the infection is going to modulate the degree of antigen presentation in class 2 abundance on these cells. Absolutely.
Right. So if you have more severe infection, it could be, more of an interferon milieu, therefore more of a Th1 response there. Really interesting. I think we're at time. I'm not sure if Annie had one other question here in terms of environment shaping effector function, capturing environmental factors that vary in inflammatory settings or in particular tissues in our in vitro experiments. Not sure if either of you could take that in the last minute. Big question.
Sure thing. I'll jump in there briefly. So I think that's one of the things that we, as a field, really need to start to look at. The idea of macrophage polarization has been around for a long time. It's a very nice, simple idea that folks latch onto. Having worked in that field for a little while now, it's far more complicated. M1, M2 does not define macrophage activation cleanly at all. And you realize that as soon as you start sequencing and running experiments with different types of modulators. So I think that, yes, it needs to be dealt with in vitro experiments and very carefully, because again, the type of modulators can even things we think are going to be subtle differences in types of cytokines can very dramatically change the FcR profile, FC receptor profiles on these cells, as well as the types of responses that they're able to mount.
Right. Really interesting. All right. Thank you, Margie and Adam. This was really insightful. Broad range of topics today: disease settings, cell perspective versus antibody perspective. So it was really interesting and enlightening. So thank you, and thank you to everyone who joined. Again, the link is in the chat. Trey put that in the chat. Apologies again for the link being off today. Hopefully we're a bit smoother at the next seminar on February 1st. So looking forward to seeing everybody there again. Thank you. Have a great day.
See you, Margie.